The transition from the test tube to the living human cell is a journey across a biological chasm, a leap from the serene simplicity of a buffered solution into the chaotic, crowded, and heavily fortified metropolis of eukaryotic life. In the laboratory experiments of 2012, the CRISPR-Cas9 system had proven its elegance in the naked light of isolation, stripping DNA strands with mathematical precision. But a human cell is not a test tube. It is a vast, compartmentalized universe, governed by a different set of rules, guarded by membranes and intricate signaling networks that have evolved over billions of years to exclude foreign proteins and silence unauthorized codes. The bacterium where CRISPR was born is a relatively open-plan structure. Its DNA floats freely in the cytoplasm, accessible, vulnerable, ready to be defended or attacked. The human cell, however, protects its genetic treasure within a vault, the nucleus. Before we continue, if this already feels soothing, you can press subscribe now and then just let the video play. This double-membraned organelle is the sanctum of the self, a restricted zone where the 3 billion letters of the human genome are kept. Furthermore, human DNA is not laid out like an open book. It is spooled, wound, and compacted into a dense substance called chromatin. It is wrapped around octamers of histone proteins like thread around a spool, then coiled again and again into fibers, creating a complex, three-dimensional architecture that renders much of the code physically inaccessible to the wandering machinery of the cell. To edit the human genome, the bacterial Cas9 protein would not only have to survive the hostile environment of the mammalian cytoplasm, but also find a way to breach the nuclear envelope, navigate the labyrinth of chromatin, and locate a specific sequence of letters amidst a library so vast that if printed it would fill a stack of books hundreds of feet high. This was the challenge that faced the scientific community as the calendar turned to 2013. Could this prokaryotic sword be taught to fight in a eukaryotic arena? In the laboratories of the Broad Institute of MIT and Harvard, and simultaneously in the labs of Harvard Medical School, researchers led by Feng Zhang and George Church were quietly engaged in the intricate engineering required to bridge this divide. They understood that the Cas9 protein, in its native bacterial form, lacked the necessary credentials to enter the mammalian nucleus. It was a molecular, undocumented immigrant. To grant it entry, they had to modify the protein's structure, stitching onto it a sequence of amino acids known as a nuclear localization signal, or NLS. This short peptide tag acts as a molecular passport. When the cell's transport machinery encounters a protein bearing this tag, it binds to it and ferries it through the nuclear pore complex, the massive, gate-guarded channels that control traffic in and out of the nucleus. This was a subtle but critical modification, a humanization of the bacterial machine. Furthermore, the language of the gene itself had to be translated. While the genetic code is universal, different organisms prefer different dialects, utilizing specific codons, triplets of nucleotides, more frequently than others to represent the same amino acid. For the bacterial Cas9 gene to be expressed efficiently by the human cellular machinery, its sequence had to be codon-optimized, rewritten to match the preferences of mammalian cells, ensuring that the ribosomes would translate it smoothly and abundantly. These were acts of synthetic diplomacy, negotiating the biological differences between a simple microbe and a complex mammal to allow the tool to function. In January 2013, the silence was broken. In back-to-back papers published in the journal Science, the teams demonstrated that the barrier had been breached. They showed that the engineered CRISPR-Cas9 system could indeed enter the human nucleus, that it could unwind the tight coils of chromatin, and that it could find and cut its target genes with the same programmed specificity seen in the test tube. The implications were immediate and electrifying. We were no longer looking at a potential tool. We were holding a functional key to the human genome. But the cut itself, the physical severing of the double helix, is only the opening act of the drama. The true magic of gene editing lies not in the destruction of DNA, but in the cell's desperate, instinctual response to that destruction. When the Cas9 scissors snap shut and the DNA strand is broken, the cell perceives a catastrophe. A double-strand break is a lethal signal. If left unrepaired, the chromosome could fragment, leading to cell death or cancerous chaos. Evolution has therefore equipped the cell with robust surveillance systems that constantly scan the genome for such breaks. Almost the instant the Cas9 enzyme retreats, the cell's repair machinery descends upon the site of the injury like a rapid response construction crew. There are two primary pathways this crew can take, and the outcome of the edit depends entirely on which path is chosen. The most common pathway is a rough, emergency fix known as non-homologous end joining NHEJ. It is a process of brute force survival. A complex of proteins, led by the ring-shaped Q70 and Q80 heterodimers, grabs the two severed ends of the DNA, trims them back to make them compatible, and then stitches them back together. It is quick, efficient, and notoriously sloppy. In the process of trimming and stitching, a few base pairs are often lost or added, random insertions or deletions, known in the trade as indels. In the context of natural evolution, these errors are usually harmful. But for the genetic engineer, this sloppiness is a weapon. If the goal is to disable a gene to knock it out, NHEJ is the perfect ally. By directing Cas9 to cut in the middle of a gene's coding sequence, the researcher invites the cell to make a messy repair. The resulting indel shifts the reading frame of the gene, turning the sensible prose of the genetic code into gibberish. The gene is effectively silenced, its function destroyed. This capability alone transformed basic research overnight, allowing scientists to systematically turn off genes in human cells to determine their function, creating a high-resolution map of genetic dependencies. But the dream of gene editing was never just to break things, it was to fix them. To cure a genetic disease like cystic fibrosis or sickle cell anemia, one cannot simply silence the gene, one must correct the specific mutation that causes the malfunction. This requires a different, more delicate pathway called homology-directed repair, HDR. This mechanism is the cell's copy-paste function. It is typically used when the cell is replicating its DNA and has a sister chromatid available to use as a template for flawless repair. The genius of the CRISPR revolution was the realization that we could hack this system by providing an artificial template. If a scientist introduces the Cas9 machinery along with a synthetic piece of DNA that looks almost identical to the sequence around the cut, but carries the desired correct sequence in the middle, the cell can be tricked. In its search for a template to repair the break, the cell's machinery may grab this synthetic strand. It aligns the homologous arms of the donor DNA with the broken ends of the chromosome and copies the sequence in between, effectively overriding the mutation with the corrected code. It is a moment of molecular sleight of hand. The cell believes it is restoring the original integrity of the genome, while in reality it is transcribing a sentence written by a human hand. However, in these early days of the revolution, a formidable challenge emerged: the balance between the two pathways. The cell prefers the quick and dirty NHEJ over the precise HDR. For every cell that successfully incorporated the correct edit, many more would simply stitch the cut back together with a scar, or worse, not repair it at all. The efficiency of writing was low, a rare event in a sea of deletions. Furthermore, a darker concern began to loom over the field, a concern that haunts every discussion of genetic intervention: specificity. The human genome is a vast wilderness of 3.2 billion base pairs. The guide RNA used to direct Cas9 is typically only 20 base pairs long. Statistically, it is possible for similar sequences to exist elsewhere in the genome, off-target sites that look enough like the target to fool the Cas9 complex. If the Cas9 acts as a guided missile, off-target effects are collateral damage. A cut in the wrong place could disable a tumor suppressor gene or activate an oncogene, potentially causing cancer years down the line. The early studies of 2013 and 2014 showed that while Cas9 was remarkably specific, it was not perfect. It could tolerate a few mismatches, binding to sequences that were slightly different from its guide and making unwanted cuts. This revelation injected a dose of sober caution, into the exuberant atmosphere of the field. It became clear that before this tool could be injected into a patient, it had to be refined. The wild-type Cas9, evolved in the bacterial trenches, was too promiscuous for the delicate clinic. It needed to be tamed. This necessity sparked a new wave of protein engineering. Scientists began to tweak the Cas9 molecule itself, altering the electrical charges of the amino acids that grip the DNA. The goal was to weaken the grip just enough so that the enzyme would only hold on and cut if the match was absolutely perfect. If there was even a single mismatch, the weakened grip would slip, and the enzyme would fall off without cutting. These high-fidelity variants represented a maturation of the technology, a move from the raw power of discovery to the nuanced precision of engineering. As the tools were sharpened, the scope of their application began to widen with breathtaking speed. It was not just about human health. In the agricultural heartlands, plant biologists realized that CRISPR offered a way to engineer crops without the stigma and regulatory burden of traditional GMO. Traditional modification involved blasting foreign DNA from bacteria or other species into the plant's genome. CRISPR, however, could be used to simply edit the plant's own genetic code, deleting a gene that made wheat susceptible to mildew or tweaking a promoter to make tomatoes grow larger. The final product contained no foreign DNA, only a native edit that could theoretically have occurred through natural mutation. It blurred the lines between nature and technology, challenging the legal and ethical definitions of what constitutes a modified organism. Simultaneously, in the realm of basic biology, researchers began to use CRISPR to construct gene drives, a concept that borders on science fiction. In sexual reproduction, a gene normally has a 50% chance of being passed to the offspring. A gene drive, however, is a selfish genetic element that forces its own inheritance. By encoding the CRISPR machinery within the genome of an organism, say a mosquito, the system can cut the partner chromosome inherited from the other parent and copy itself into the break. This ensures that nearly 100% of the offspring inherit the modification. Driven by this mechanism, a genetic trait, such as infertility or resistance to the malaria parasite, could sweep through an entire wild population in a handful of generations. It offered the tantalizing possibility of wiping out vector-borne diseases that kill millions, but it also carried the terrifying risk of ecologically irreversible changes, of altering entire species in the wild with no undo button. These ripples of possibility spread outward from the nucleus, touching every aspect of the biological sciences. But to truly understand the gravity of the edit, one must return to the microscopic scale, to the physical reality of the DNA molecule itself. DNA is not an abstraction, it is a physical object, a long, viscous chain of hydrated phosphate and sugar. It has stiffness, it has tension. When Cas9 binds to it, it forcibly unwinds the double helix, bending the DNA, probing for the PAM sequence, the protospacer adjacent motif that serves as the landing lights for the enzyme. This physical interaction is a dance of atomic forces, of hydrogen bonds breaking and reforming, of electrostatic attractions pulling the protein into the major groove of the helix. The visualization of this process, captured through techniques like cryo-electron microscopy and high-speed atomic force microscopy, revealed a machine of startling structural beauty. The Cas9 protein looks like a clam, composed of two main lobes, a recognition lobe that holds the guide RNA and a nuclease lobe that contains the cutting domains. When it encounters its target, the protein undergoes a massive conformational change. It shifts, rearranges, and locks onto the DNA, creating a R-loop where the RNA guide hybridizes with the DNA strand. It is a kinetic, dynamic event, a molecular embrace that seals the fate of the genetic sequence. This physical intimacy between the engineer's tool and the fabric of life is what makes the technology so profound. We are not interacting with life from a distance. We are interposing our intentions into the very molecular handshake of existence. And as the technology moved from the academic bench to the commercial sector, the question of ownership arose. Who owns the rights to this fundamental mechanism of nature? The patent dispute that erupted between the Berkeley team, Doudna/Charpentier, and the Broad Institute team, Zhang, was more than a legal squabble. It was a symptom of the immense value, both financial and humanitarian, that society placed on the ability to rewrite the code. But while lawyers argued over priority dates and lab notebooks, the science refused to wait. It flowed around the legal barriers, driven by a global community of researchers who saw in CRISPR a way to tackle the most intractable problems of biology. By 2015, the CRISPR zoo was expanding. Scientists were not content with just Cas9. They went back to the microbial well, mining the genomes of other bacteria to find new versions of the system. They found Cas12a, formerly CPF1, a leaner, smaller enzyme that cuts DNA in a staggered pattern, leaving sticky ends that are useful for certain types of insertions. They found Cas13, which targets RNA instead of DNA, offering a way to transiently modulate gene expression without permanently altering the genome, a temporary mute button rather than a permanent deletion. The toolkit was becoming a toolbox, a diverse collection of molecular instruments, each with its own strengths and specializations. Yet amidst this explosion of diversity, the central narrative remained focused on the ultimate prize, the therapeutic correction of human disease. The concept of somatic gene editing the cells of a living patient to cure a condition, began to move from theory to preclinical reality. The target was often the blood. Blood cells are accessible. They can be removed from the body, edited in a dish, and returned to the patient. This ex vivo approach became the proving ground for the first therapies. Conditions like beta thalassemia and sickle cell disease, caused by single-letter mutations affecting hemoglobin, were the low-hanging fruit. The strategy was elegant in its circumvention. Instead of fixing the broken adult hemoglobin gene, which is difficult, scientists used CRISPR to break a break gene, BCL11A, that suppresses the production of fetal hemoglobin. By breaking the break, they could reactivate the fetal hemoglobin, which naturally fades after birth, providing a functional substitute for the defective adult protein. It was a bank shot, a clever biological workaround that relied on breaking a gene to fix a disease. But as we look toward the future, toward the idea of editing solid tissues, muscles, hearts, brains, the challenge of delivery returned. How do you get the Cas9 protein into a heart-muscle cell buried deep within the chest? You cannot take the heart out and put it back. You need a vehicle. The field turned to viruses once again, this time not as enemies but as mules. Adeno-associated viruses, OVs, were engineered to carry the genetic blueprints for CRISPR, stripped of their own pathogenic genes. These viral vectors could be injected into the bloodstream, homing in on specific tissues to deliver the editing machinery. It was a poetic inversion, using a virus to deliver a bacterial immune system into a human to cure a genetic defect. The biological loops were closing, the boundaries between viral, bacterial, and human biology becoming increasingly porous in the hands of the synthetic biologist. As Part 2 draws to a close, we stand in the mid-2010s, a time of feverish optimization and expanding horizons. The tool works. It works in human cells. It works in animals. It works in plants. We have learned to control the cut, to improve the fidelity, and to deliver the payload. But the greatest threshold remains uncrossed. All these edits were somatic, changes made to the body of an individual that die with them. The germline, the sperm, the egg, the embryo remained a line in the sand, a boundary guarded by heavy ethical caution. To edit the germline is to edit the species, to introduce changes that will echo down through every subsequent generation until the end of the human line. The technical capability to do so was arguably already in hand. The question was no longer about the science, it was about the soul of the species.